Okay. Good afternoon. We will start the second lecture on homologous recombination. So in the last class, we discussed about rec-BCD pathway in E. coli. So the objective being that the cell or bacterium here will be bombarded or sometimes is required for energy to depend on DNA from the external sources. If that is the case, how is the DNA used up? And it is also involved with repair mechanisms. If a bacterium has endured mutations, such as in the case of SOS response, for example, there is a way to repair it from the DNA that is coming from the outside, right? Exogenous DNA. And that DNA can be from other E. coli, that is dead E. coli, or it could be from some other organisms. Every organism has DNA. So if it has to undergo homologous recombination, there should be a way of somehow ensuring that the DNA that is coming from the outside is that of E. coli, right? What it integrates into its genome should be E. coli should have a mechanism to distinguish between what is E. coli DNA and what is not E. coli DNA. So it depends on something called as Chi sites. The abbreviation of full form is Chi, C-H-I, that is crossover hotspot instigator, CHI. Those are some sequences that are present in E. coli in numerous places. So based on that, they increase the frequency of recombination, that is homologous recombination. So in the rec-BCD pathway, we have several proteins. As you have to remember always the aligning of DNA, processing of the ends, strand invasion, branch migration, and resolution. Those are the steps that are involved in homologous recombination. When we speak of real examples, there will be some variation to them. Okay. Rec-BCD are Rec-B, Rec-C, Rec-D are proteins. They form a multi-subunit complex, which are helicases as well as nucleases, right? Say, for example, the DNA that has come from outside should be processed. If it is not having Chi sequence, that means it is not E. coli. But if it has a Chi sequence, that means it is likely or more probable. It's not with absolute certainty, but you can say it's probable that it is likely from E. coli. So rec-BCD proteins are the proteins that will degrade the incoming DNA, but if it encounters a Chi sequence, like in this case, that strand will not be broken, and you'll have the other strand broken by these proteins. And then there will be strand invasion like step, and that is involving a protein called Rec-A, right? Rec-A is a protein which we already discussed in SOS response. It is single strand binding specific, and it has some properties of strand exchange. We have seen that also that it binds to the protein, the single strand DNA in this fashion, more towards the 3' end, and it also does strand exchange. How does it do that? It first binds to one of the strands like this and coats that protein as a filament. What you're seeing here is a cross section, and then it binds to a second double-stranded DNA, and it will check for homology. Wherever homology is good, then the strands will start base pairing. Exchange will happen, which will result in something like this. And for resolution, we have a step called as, for branch migration, that is mediated by proteins RUV-AB complex. RUV-A is the one that is important for recognizing holiday junction, RUV-B for branch migration. It is like coupling two RUV-B proteins or protein complexes. They rotate in different directions. As a result, DNA will move, the branch will move in one of the directions. Then the RUVC is the protein that is involved in resolution of the holiday junctions. And it can happen in site 1 or site 2. There is a modest sequence specificity. It's not randomly anywhere it would happen. And RUVC is the protein that makes these cuts, and then these will be joined. That is how holiday junctions are resolved. We also learned about what happens during meiosis. This is what you should remember. Homologous chromosome pairing will happen during meiosis. You have a formation of tetrad where homologous chromosomes come together, and there will be recombination between non-sister chromatids, like in this case, if you can see it. And there will be exchange of fragments of chromosome. And eventually you will have the generation of recombinant chromosomes. The outside ones, the completely blue and completely red ones, are not recombinants because they haven't had any informational exchange. Whereas between the ones that I'm pointing now, there is a recombination that has happened. And that is how meiosis generates variation, or there is recombination happening. Okay. And that is mediated by how the double strand break happens is dependent on spole 11, and they act similar to that of topoisomerases. So there are certain sequences, spole 11 cleavage sites, and then they cleave at those sites, and then exchange of DNA happens. So there are proteins called as MRX. Here also it is much more clearer to see the double strand break model here. Spore 11 makes a double strand cut, and MRX proteins also integrate, and they process the DNA to generate 3' overhangs. These 3' overhangs will be bound by DMC1 and RAD51, and they will allow the strand invasion to happen. And then you will have polymerase filling up these gaps. And at one point, they will be resolved. So in today's class, what we are going to discuss is mating type switching in yeast, and the other concept that we will learn today is gene conversion. Gene conversion is just a name, but we will know what it actually means. It's simple. So mating type switching is something, if you have watched the animation, you should have had a good amount of idea by now. So as I was telling you in the last class, I was introducing you, there are two, so yeast in this case can undergo reproduction asexually and sexually. Asexually by binary fission or budding, actually. It will bud and it will keep giving rise to more and more cells. Here is A cell, A-type cell, and the other one is called as alpha type. Okay, there are two sex types, you can call it, or mating types, if not sex. You call it as A type, A mating type, and alpha mating type. So here is a depiction of a picture which says A will give rise to a bud and will form another A cell here. So you have, that is a sexual reproduction. But sometimes one of the cells can switch its mating type to alpha. Okay. I didn't bring the pen, but we'll adjust with what we can have. Okay. So how does that mating type switching happens is what we'll try to understand. Again, in this case too, we know a lot of details, but there are a lot more details that we don't know. So we are pushing our knowledge to the edge of what is known and unknown. It's a good way to learn things. So while you are learning it, know that some things we don't know how they actually happen. There is still some more research to happen, but it's a good idea to understand the nature of nature itself, how things happen in the nature. So here is another picture depicting the same thing. Alpha, it gives rise to alpha cells, but occasionally they may switch and give rise to A-type cells. And A-type cells will keep running asexually, and they may switch to alpha type. During maybe some sexual reproduction will require the fusion of alpha and A. Anytime there is a alpha cell will not fuse with another alpha cell for sexual reproduction. It will only fuse with A cell. Okay. So if we had one cell of alpha and we allowed it to grow, at one point, it will automatically, the A-type cells will come. And how does that happen? It is because of this mating type switching. And sexual reproduction can only happen between A and alpha. I'll try to include a picture at a later time, and I'll show you that in the next class maybe. So in this, yeah, if you can understand this, this is how sexual reproduction happens. You don't need to know all these things. This is just for understanding. But sexual reproduction happens between A and alpha. So A produces a certain components that will bind to alpha, and alpha produces something that will bind to A. Okay, receptors and signals. There are many things. Right now, you don't have to bother about it at this time, but you can always Google it up and learn. Then these two cells will fuse and make a diploid cell. Until then, the yeast there was haploid in nature. Now you have diploid cell. And this cell eventually will again form a diploid cell from which you will get again meiosis, and then you will have haploid cells. Okay. So why should this happen is always an interesting question. Why do we need sexual reproduction, or why does sexual reproduction arise in every type of organisms? In yeast, in the simplest form, you're seeing here mating type switching. It's kind of sexual reproduction. Two differentiated gametes fusing to give rise to something. Same thing you'll see in animals, mammals, snakes, or you can find even plants. Even the flower itself has two sexes. You can take earthworm. You have two sexes. They are hermaphrodites. So why should sexual reproduction arise in all these things is a mystery that people are working on. I am also interested in that, and I have my own theories, but we'll get to it later. I hope you should be inquisitive about this, because it borders between what is science, biology, and philosophy. Philosophy is trying to know the truth. Why does it exist? It doesn't matter what others have told you. It doesn't matter what you have read. You can still ponder upon it and try to get into your own answers, but they should be justified and maybe malleable, in the sense that you should be able to change your opinions when there is new evidence. Or you can also accept, maybe I know till this much, I don't know further than that. That is also a good stance to take in science and philosophy. So now I'm trying to describe how a A-type cell changes to alpha-type cell, or alpha-type cell changes to A-type cell. Okay, so let's just see the genetic architecture of that region. In that place, what you're seeing is, it is called as, there is a mat locus. So there are two types, mating types. There is mat locus. This is mat locus. This on the other side that you're seeing is called as HML, and the other one on the other side you have it as HMR. HML is hidden mat locus. HMR is hidden mat locus. Okay, on the right side you call it as HMR. On the left side you call it as HML. Okay, hidden mat locus. The one active mat locus is in the middle of it. This is the one that I'm trying to show you. So on the left locus, you have the genetic information to have alpha type. On the right side, you see here, it has genes for A mating type. Okay, HML alpha, HML A. If you remember it that way, you know what genes are in what. But as the name suggests, that they are hidden, in the sense that they are not expressed, they're silent. You can also see it here. It is called as silent locus. Locus means it's a region. That is what is said in here. In genetics, you just use locus or loci for plural. So whichever, if there is a cell like in the yeast, it has both genes for alpha as well as A, but only one of it is expressed, and that expressed locus is called the mat locus. Okay, and that is the only one that is expressed. So whichever genes are here, in this instance you have A type genes, so the cell is A type cell because those genes are expressed. If by some reason you have the alpha sequence here, alpha, then that cell, because the alpha genes are expressed here, then that cell will be alpha type, alpha mating type. I hope you understood. I'll try to quickly review that again. So there are three regions in all yeast cells. Here is the mat locus, that is the only expressed of those three. You have HML alpha, that is hidden mat left, which has the alpha genes, but it's not expressed. And you have HMR A, which is also not expressed. Those genes are A. The only expressed region is called as mat locus, mat. And whichever genes are present, if A genes are here, then that cell will be A type. If alpha genes are in this location, then that cell will be alpha type. The question what we are trying to know now is, how does a cell that is A type change to alpha, or switch to alpha type, or alpha type switch to A type? Here is the molecular mechanism. It involves homologous recombination, and that is the reason why we are studying it. Okay, so I hope you can recollect that mat locus. You have to keep this map in your mind. Okay, mat locus is here, HML alpha and HMR A are on these sides. So there is homologous recombination. This DNA can bend like this, and homologous recombination happens between alpha and mat. If it happens that way, then the A type cell will become alpha type cell. If there is already alpha, homologous recombination can happen between this region, that is the mat locus, and the HMRA, and then the cell will switch from alpha to A type mating, A mating type. So how does that at molecular level, how it happens is what is our concern. So the first thing is, so we are assuming the cell is having mat alpha, which means the cell is actually in alpha, because mat is the only one that is being expressed. So there is a protein called HO endonuclease, which will cleave the DNA here. That happens in the mat locus. Okay, so the alpha region, at one of the borders, you have the cleavage like this. And then this is processed, that is 5' resection, which means the 5' DNA parts are broken down. This region was broken down, and also here. This will allow the generation of 3' strands. So one of these 3' you have here, it will make strand invasion to the top, or the HMR A region like this. And here you have a replication fork, right? And you have the DNA polymerase will keep synthesizing it this way. And now we are in this stage here. So one of the strand is completely synthesized. What is this dark green indicating? Dark green is the genetic information for A, right? It is for A. And then that strand that is been, one second, I will try to see. Yeah, the strand that is synthesized here will now move to the bottom, like is shown in here. And that is used as template, and new DNA is being synthesized here. The alpha sequence here, it is broken down and removed out. Okay, that is how compare what is here and compare with that of what is around in this case. What you'll see is in A stage A you had HMR A, and at the mat locus you had alpha information. But by the time we came to A is F state, you can see that HMRA remains the same, of course, and mat became from alpha to A information. And now this will be expressed, and the alpha cell will now become A cell, A type, A mating type. I'll quickly review again. The endonuclease, HO endonuclease, will make the double strand break. And there are several proteins that could be involved in removing the 5' ends so that 3' overhangs are developed. That 3' overhang now will make a strand invasion to the HMRA, and that information, the DNA polymerase will copy that sequence information of A, and then it will bring it down to this strand. And the other alpha strand that is present here will be excised and will be removed away. And now what you have is a new synthesis of DNA strand, and that is how two strands of A information is made at the mat A locus, that is the only expressed locus. Okay, this is called as mating type switching. Why should it switch, and why should sexual reproduction happen is a question that you can ask. It might be more for you at this level, but it's always important to be curious and try to find out your own answers. There are a lot of research papers on these topics you can try to explore. So the next question, next topic of interest today is gene conversion, and I hope you will try to understand these. In many cases, we have read about heteroduplex, right? Means mismatches. You have A, and in heteroduplex, I said recombination or homologous recombination is fruitful if only it happens between two DNA molecules that are highly similar but not identical. That means there should be at least one base difference between them. And if that happens and homologous recombination has occurred, what you will end up is with mismatches like these. And sometimes you can also have large number of mismatches, or sometimes you may have insertions and so on, or deletions as well. In this case, DNA should be repaired. This is A base pairing with G is not normal within DNA. It is not correct. And that should be repaired. And that has to do or results in conversion. And that's what we will learn now. So you remember recombination happens, right? We spoke about something capital A. These two are different strands of the same DNA molecule, two strands, and here you have two strands. We also spoke about some recombination will always occur between non-sister chromatids, say. And when recombination happens between these two, then you will find mismatches like this, right? Capital A, small a. They're here in this case, they're indicating genes at the level of genes. If I have to write, as I was telling you in the previous slide, I was telling you that there is a mismatch, capital small a, sorry, adenine base pairing with G, guanine. That's not right. So that is a mismatch. So if you had A and G, what is going to happen? Say, for example, the top strand is coming from a gene that is A. Okay, that will be confusing, I guess. Maybe I'll leave it here and we will get back to it if there is a problem. So this DNA has capital A, both of them, the information for capital A. This DNA has information for small a. So this will be 25%. After what do you say? After meiosis, you get four sperm or four gametes if it is in spermatogenesis. So you have four cells are forming: 1, 2, 3, and 4. So there is no difference in this, in the top one, and there is no difference. These are non-recombinants. They did not recombine with anything. Recombination has happened between these two, right? So you have a probability of, this is capital A and this is small a, genetic information, no difference at all. What will happen to them is the problem. During recombination, you will end up forming a heteroduplex like this. And either this capital A will be repaired or the small a will be repaired, right? Because if it is mismatched, that is what happens. Like we read about mismatch in prokaryotes, we have a way of determining, and especially if it is going along with the replication, we can figure out which one is the parental strand and which one is the newly synthesized strand. The same way we can also in eukaryotes too, we have basing on interaction with pCNA or sliding clamp and Okazaki fragments, mismatch repair can detect what is happening. But in this case, in recombination, it is not a whole cell replication, isn't it? It is rather some mixing of DNA. And mismatch repair may have difficulty recognizing which one is parental strand and which one is not. So in that situation, there is a probability of the capital A being repaired, or there is equal probability of small a being repaired. If the small a is repaired and copied, the capital A information is copied downwards, what will you get? You have capital A and small a, those are non-recombinants. And if they were repaired, what you are getting is two more capital A's. What would you normally expect? You would actually expect two capital A and two small a. But that's not what is going to happen. You will either get three capital A's and one small a, or three small a's and one capital A. And what will happen is dependent on the probability of which of this heteroduplex is being repaired by mismatch repair. Okay, if the capital A is repaired on the top strand, is removed and small a information is copied, then it becomes the information of small a. That is gene conversion. Okay, it's just a name. It's don't get carried away with that a gene is present in a cell and is getting changed away. That's not how it is. It has to do with recombination and followed by repair. Homologous recombination followed by repair. Okay, it's just the change in the frequency of what is happening. Either you'll find this way or you will find it in this way, and that is because of gene conversion, a phenomenon called gene conversion. Okay, I will not take this. This is not included in your or you'll need to visualize it better, but it's all right, you can skip that. So that brings us to the end of our chapter on homologous recombination. Typically, these will be the questions will look something like these. These are the exactly what I have discussed: double strand break model, rec-BCD model, meiotic recombination, and mating type switching in Saccharomyces cerevisiae, and also gene conversion. Okay, that is what that brings us to the end of this class today.